One widely known fact about Emma is that she's our lab's self-appointed social chair. I think she might also be the Department of Human Genetics social chair somehow.
I'm roommates with the
Roommates.
social chair of human genetics. It rubs off.
But a lesser known fact, maybe, is that Emma is the cultural ambassador for Minnesota. She brings exotic treats to the fourth floor, including things like Snicker salad, which is a Midwestern salad that doesn't have any vegetables in it. This one at least has apples, so that's like, it's got something going for it.
Cellulose.
Emma joined our lab from Mike Travisano's group at what Emma calls the real U of M, so we should probably fail her for that. Where she studied multicellularity in yeast. And despite our lab's strict no eukaryote policy, Emma's managed to start asking questions about multicellularity again, but using microbes. So I'm excited to see what this is.
Do you like my eukaryotic shirt?
Oh, minus 3 points.
Worth it.
Okie doke, welcome to my talk. Normally I'm only up here for about 10 seconds, so it's kind of exciting that I get your attention for the whole session. Okay, so starting my talk, I want to talk about what this talk is not. And what this talk is not is another entry into sort of the niche construction discourse of the 20-aughts and the 20-teens. So if you came here for a fight, we'll save that for the bars. Okay, so what I'm trying to do is I'm not trying to state that niche construction or niche construction is sort of this universal, all-encompassing idea that applies to everything from bacteria to snakes, but instead that it's a way to organize ideas and approach problems within the specific system that I'm working with. So there's going to be 2 main parts to this talk. The first part, I'm going to build intuition for host-parasite coevolution and for niche co-construction. And then for the second part, we're going to start to explore the open questions sort of in this area using the ideas we build in part 1. So let's get ahead on part 1. If you don't know anything about host-parasite coevolution, then let's start at the very beginning. So the host in general is sort of what we think of as the larger organism, and the parasite is the smaller organism. So the parasite lives on or in the host, and basically takes some resource or has some sort of negative effect on the host. So we end up getting a benefit to the parasite and a negative to the host. The parasite isn't just doing this for fun, but is replicating within this host, and then is dispersed, sort of expelled out of the host and dispersed to find new hosts. This can end sort of one of 2 ways in humans. 1, maybe the parasite takes a little bit too much and we're having a little bit of a sad day. Or 2, you go to the hospital and you're getting that figured out. So what I'm looking at is not sort of the human interaction, but how we can look at this host-parasite coevolution, modeling it in a bacteria system. So bacteria is our host, our bigger organism, and our parasite is our bacteriophage, or phage. I'll probably refer to it as phage for most of this talk, because it's a little easier to say. Basically what this happens is this is a little viral parasite that jumps on, basically attaches to the bacteria, injects its genetic code into the bacteria and takes over. So it takes over all that cellular machinery and those resources, and it basically gets that positive, takes over the bacteria. Well, the bacteria is no longer getting to do its little bacteria life, so it's not having a great day. So much like in our example earlier, the phage replicates inside the bacteria and bursts out. So this is a highly simplified depiction of what's actually happening, but it sort of paints a general picture about the sort of the interactions that we're going to look at today. So the typical relationship ends just in sort of this bacterial death. There is sort of like this whole world of bacterial immunity and phage response, but that's outside the scope of this talk, but it's super interesting. And then also we're going to just focus on lytic phages. So basically this idea that you attach, you inject, you replicate, and you burst. So why would we study this when we have a whole bunch of perfectly good humans to study? Well, 1, it's deeply unethical to infect a human with a parasite. It's also wrong. It's not just unethical. You shouldn't do that. Also, humans famously don't fit very well in the negative 80-degree freezer. So if you want to sort of save them and sequence them later, it's a lot of space issue. So bacteria, we have rapid generation times. We've established reference genomes. They're cheap and easy to sequence. easy to freeze, and also we have the benefit of them being very clinically relevant. So we can sort of establish this sort of ecological, evolutionary relationship, but then also use it to push forward some human technologies. So what does a typical bacteria and bacteriophage research setup look like? The most general sort of thing is you have a series of test tubes filled with media, you throw some hosts in there, you throw some parasites in there, and you see what happens. And this generally takes place in something we call a well-mixed culture. So a well-mixed culture, as the name suggests, is we are mixing. We are putting it into the incubator and shaking it up. This keeps the culture oxygenated, it keeps things moving around, and it basically gives us this sort of like homogeneous situation. So at the end of this example, we see that our sort of our standard control tube just has bacteria in it, which is great. It means that we didn't contaminate it. And our second tube, we see that we have no more bacteria left. We only have the phage, meaning that this phage figured out a way to basically infect all of the hosts and use up its host population. And then in the third tube, we see an example where the bacteria are here, but with no phage. So the bacteria found a way to stay resistant and basically outlive the phage to extinction. So we see a lot of sort of the idea of we have winners here. So what if we don't mix it up? What if we leave it static? Something really interesting happens when you let bacteria grow in static culture. You end up seeing these sort of gooey clumps form at the top. And this is called a biofilm. And as Luis mentioned, I have a soft spot for cellular conglomerations. So I really, really, really loved biofilms when I first started reading about them. But the issue I had is host-parasite coevolution in bacteria and phage in a well-mixed culture. You have so many papers. But when you're looking for this interaction and how it changes in biofilms, suddenly I had a lot less answers to my questions. So I want to know, basically, what's happening in these biofilms. So what is a biofilm, physically? A bacteria basically finds a surface, a bunch of cool cell signaling stuff changes, and we end up seeing the promotion of this extracellular polymeric substance. This is generally sort of a vague term purposefully because this is highly variable depending on the strain, depending on the environment, on what this sort of goo looks like. But basically, you can think of this as like the snot ball at the bottom of your shower when you haven't cleaned it in a while. It's that. Okay, so I know, delicious, right, for lunch seminar? Okay, so inside of that snot ball, we have a bunch of cells. Obviously, those are our friends that are producing this sort of extracellular polymeric substance. And then we also have extracellular DNA, which is very cool. It comes from both the cells that die in the actual biofilm and from the environment. And this acts as a strengthening agent for the biofilm. So you end up kind of like rebar in concrete. You see this acting as sort of a structural component. We also see a variety of proteins, sugars, and lipids. This is also left intentionally vague because this differs wildly depending on the type of microbe that's in these and the environment itself. Lastly, this is a highly, highly hydrated system. Over 70% of this biofilm, any biofilm, is basically water. So you see these H bonds of these hydrophilic polysaccharides in this EPS, basically holding on to water. You see water channels, you see water pockets, and these are going to become relevant. So despite my beautiful cartoon, this is sort of a more accurate depiction of a biofilm. This is a scanning electron microscopy image of a biofilm, and we can see that this isn't like one consistently sort of distributed situation. We see cell clustering, we see more of this sort of like dried polysaccharide business. So it's a highly, highly variable situation. And one thing before we go on is I want to emphasize that 80% of prokaryotic cells live in this biofilm form. So the most likely way you're going to run into a bacteria is within these biofilms. Okay, so what is a biofilm ecologically? A niche. It's the range of resources and conditions that allow the species, the bacteria, to maintain their viable population. But more interesting, it's an example of niche construction. So I'm pretty sure everybody is familiar with a beaver. A beaver is a great example of niche construction where it modifies its environment, changing the selection pressures on itself and also changing selection pressures of other things in the environment. So we see the building of a dam, which basically protects the beaver. It now has a house for itself and its young, but it also might flood a river. It stops the water flow in some places, and it exposes a whole host of organisms to new selective pressures. So if we imagine a bacteria doing the same thing, we see that these bacteria find a nice little location to settle down. They build their EPS house, and they are now changing both the physical space and the chemical environment in which they are now located. So what does a biofilm provide other than just sort of a nice little place for them to hang out? One of the big things is getting back to that highly hydrated situation. It's protection from desiccation. So if you're a bacteria, you're really likely to dry out if you get left in the sun. But if you have this sort of jelly layer protective situation going on, you're less likely to do that. You also reduce toxin exposure. So you're decreasing sort of the amount of things that can access you immediately. Maybe they're slowed, maybe other cells die first. You're basically just slowing down that selection pressure. And third, you're blocking phage from entering and getting directly to you, kind of. So this is one of the first lies I am telling in this presentation, because previously a lot of people thought that biofilms were sort of this end-all be-all of phage blockade. But actually, there's more to that. Otherwise, I wouldn't really have a talk. Okay, so phage can and do get in. One way they get in is through water channels that I mentioned earlier. So they can diffuse through these water channels and get into cells that way. And then some have evolved to have basically these little enzymes on their tails. A phage tail is like these little feet. They have a little, think of it as like a little spiked booty that they're able to sort of like kick their way through the EPS. So, are they changing the biofilm structure? Yes, in many ways. And we're going to talk about sort of the ways that this is happening. So one, phage can change the amount of biofilm produced. They can change the biofilm composition. And what I'm proposing is that phage can change the actual structure of the EPS itself. So we're going to go through each one of these individually. So phage can change biofilm production. These are two beautiful black nylon beads that I grew biofilms on. So these are some of those sort of snotty biofilms that I'm obsessed with in lab. So this happened over basically, you can put this bead in the culture. It has a little bit of texture to it, so the bacteria stick to the bead. And then each day you can move the bead to a new culture, and only the cells that are best at sticking to the bead get moved. So you're selecting on the ability to form an extra sticky, extra good biofilm. So I evolved a good biofilm former, and then I grew it in the bottom of in a 96 well plate, dumped out all of the planktonic cells, so all those free floaters, and then dried and stained the biofilm chunks. So anything that you see of these little black chunks is biofilm. So this is one of the high biofilm production cell lines. So we get this like very impressive chunk situation going on. But what if we had grown it with phage? So at the same time that this was being grown, on the other side of the plate, I had the exact same concentrations, but I added different titers, different amounts of phage. And what we end up seeing is an incredible increase in biofilm production. So this is from a random bacterial cell strain that I pulled from the freezer and a phage strain that I knew could infect it, but I didn't know really anything about their evolutionary history or if this is a biofilm trigger. These were sort of just two randomly selected things. And within 24 hours of this co-culturing, we end up with this incredible surplus of biofilm formation. So we know something is happening when we apply phage to a biofilm. So they can also change the biofilm composition. So in this case, we see that in this Fernandez et al. Paper, they grew biofilms and then did confocal scanning laser microscopy, and they got these sort of beautiful biofilms where it's clusters of cells in these little island shapes. And these cells, you see the green are the live cells and the red are the dead. So basically there's these wonderful colonies of living bacteria sort of on their dead relatives, on a bed of both dead cells and extracellular DNA. So this was grown without phage. When the same biofilm was grown in the presence of phage, we see suddenly these islands aren't so empty anymore. We see that all of a sudden this middle area is filled with dead cells and extracellular DNA. So when they did tests on these, they saw that those higher eDNA concentrations were more stable and could better withstand physical disturbance. So these biofilms that had been predated by phage, that had been attacked by these viruses, actually ended up stronger than the non-attacked biofilms. So we know that phage can lead to modifications of this matrix, of this biofilm, by directly acting with the bacteria. So by either this could either be a rapid selection instance, or it could be a plastic response. It really depends on the strain. Or we have this actual lysis of cells resulting in this surplus of extracellular DNA. So what I'm interested in is can phage modify the EPS directly? Can we get this matrix modification not by interacting with the bacteria, but interacting with that goo? So my proposed hypothesis is the idea that we have this biofilm, and then we have, say, like a toxin, say an antibiotic, and we're applying the antibiotic to the biofilm. This then is able to get sort of through these water channels, just like a phage could. But unlike phage, they can't replicate. This is sort of once it gets in, it's all that it is. So maybe we lose a few cells to antibiotics, but then we encounter a resistant cell. This cell can then spread, and then suddenly we are left with a biofilm that is far more antibiotic resistant than it was at the start. But what if we had phage in there first? So if we have water channels where phage can get through, or one of these phage with a D polymerase that can sort of eat through the matrix, well, how is that going to change the selection pressure? So suddenly we have a lot more space within our biofilm. And if we apply those same antibiotics, do we see this deeper penetration of antibiotics into the biofilm than we did with just the normal antibiotic application? We don't know. That's what I'm hoping to convince you of today. So these are the main three ways. These two obviously have been shown. And then this as a corresponding sort of facet of the ways that modifying a niche can affect the coevolutionary process. Okay, so to put this all together once again, I'm saying that bacteria can create biofilm, which is an example of modifying a niche by producing EPS. In turn, the production of this EPS changes selection pressures on phage. These phage are now selected for their ability to get through this goo and to the host. The phage, in turn, modify the niche by changing the internal structure, by leaving pockets of dead cells once they get further in, or by physically degrading using that D polymerase, which in turn changes the selection due to the modification of the niche, now letting different environmental factors in. Again, this is a proposed thing. I'm happy to be proved wrong, but I look forward to figuring it out. Okay, we've made it to part 2. Now we're going to start asking some questions. Let's use this framework first to see what the literature already has for us. So I'm interested in to what extent does the existing bacteriophage literature show evidence for niche co-construction? And when I'm talking about the literature, I'm talking specifically about biofilm literature that has phage parasitism. So can we view this current interaction sort of through the lens of niche co-construction? So the first thing that I need to do to figure this out is I need to figure out what traits actually count as niche co-construction and if we can find them in the literature. So in general, I've sort of divided this into 3 categories: changes in biofilm structure, so biofilm size, the number of viable cells within, and the composition in general, that amount of extracellular DNA, cell densities, and matrix architecture. Additionally, changes in bacterial strategies is something I want to look at. So not only just EPS production, but also what other general environmental responses are they having. And then same thing with phage. I want to look at EPS degradation specifically, but then also does diffusion change, does burst size change, do all of these different sort of phage traits that we think about in well-mixed culture change in these biofilms. And this is a living list. I'm not married to the idea that these are the end-all be-all of phage niche co-construction. This is more of the framework to start digging in and seeing if we can find this relationship already in these papers. So, looking at 5 randomly selected phage and biofilm papers, biofilm quantity was measured in 5 of them. Biofilm viable cell counts was only found in 2. Qualitative cell distributions were found in 4, but these were done in a sort of pseudo, like, this has more and this has less situation, not actual, like, quantitative numbers attached to that. Changes in bacterial resistance was only looked at in one paper, and no mechanisms were investigated, just if bacteria host range changed, or phage host range changed. And one thing again, another lie that I'm telling is this was not necessarily a random selection of papers. These papers, their goal was all to look at the ability of phage to eradicate a biofilm. So the reason people are interested in this is because of phage therapy. So this is one of those medical technologies that I alluded to earlier. So phage therapy is basically the idea of, can we use phage instead of antibiotics to kill bacteria? This could be both in sort of like an industry context or in a medical context. And the rise in interest in phage therapy has resulted in more labs getting into this idea of host-parasite coevolution with phage. We see really sort of interesting divides between the style of lab. So what I want to do, what this has led me to, is to try to characterize the differences in niche co-construction traits between sort of these eco-evo and medical subfields. So I tried to find an emoji representing all of the labs in this department, but Microsoft Office does not have like a good statistical spatial analysis emoji. Maybe next time, Gideon. Okay, so let's talk about what the main differences are. So the main differences that I've seen so far is that in the eco-evo world, you see a lot of defanged, sort of like safe to use model bacteria. Cheap and cheerful, so to speak. E. coli, P. fluorescens are the big ones. You see them grown on very common lab substrates like microtiter plates, glass slides, glass beads, and you see coevolution motivated. So you're looking at a lot of sort of the specific switching of alleles. In the medical world, it looks a lot different. You end up with these focused on something called escape pathogens. These are the top sort of selected, most sort of urgent pathogens that we think are going to gain full antibiotic resistance in a medical context very soon. A lot of them use medically relevant substrates like porcine skin, catheter tubing, and various implantable device materials. And you see them largely eradication motivated. So a lot of these papers, the end-all be-all goal is to get rid of the biofilm, and they don't necessarily look at what's happening in the process. So what will we learn from this? So what I want to do, what I actually want to get out of this is, one, I want to look at the literature see what we might be missing by not taking a niche co-construction approach. I also want to look at the methodological differences in eco-evo and clinical biofilm phage research. Both 1 and 2 basically build to 3. I want to figure out what have we been missing sort of by not borrowing from one another, and also what are we missing by just the pure types of methods we're using. Is studying eradication and not paying attention to the ecologically and evolutionarily motivated factors resulting in sort of less information than we actually could be getting? I think there's a lot of good overlap between these, but I think that this really could benefit from more integration. And hopefully the next 2 experiments that I'm going to tell you about, that I'm setting up, will combine elements of both of these sort of methods to then create a more holistic approach. And then this work is largely, I did not invent, obviously, the concept of evolutionary medicine. This was largely motivated inspired by reading papers from the Cooper Lab and the Center for Evolutionary Medicine at Pitt. Super, super, super cool work. Okay, so the next big question, now that we've sort of looked into the literature and figured out our gaps and sort of what we could be studying better, I want to figure out how phage characteristics impact biofilm co-construction and host parasite evolutionary strategies, which is kind of a mouthful. So this is a little bit more of my EEB themed question. To understand if we have this sort of changing in environmental selection pressures as a result of phage modification of the niche, we first need to figure out if phage are actually modifying the niche in a way that matters. Okay. So to start this, I need to pick a system. So my host parasite choice, I'm going with the good old classic E. coli, but to make it sort of a medical crossover event and also convenient for host parasite coevolution, we have the E. coli K1 serotype. K1 basically just means it produces the K1 antigen on the capsule, which is what this sort of halo represents. And the capsule goes on to be a big component of the EPS of that biofilm matrix. And it's also super clinically relevant when it comes to things like bacteremia, septicemia, and neonatal meningitis. So what phage am I going to pair with this? We have a really sort of convenient answer. We're going to use a T7 phage, or T7-like phage, and it has to have a few key components. The first component is it needs to have functional genetic code. This seems pretty obvious, but also it needs to have functional genetic code that I can knock out easily. And this phage lends itself to that process, as you can hit it with a little bit of UV, and you can keep the phage intact, but get rid of its ability to replicate. And hold on to that thought. It also needs to have a depolymerase, and this phage, very conveniently, has a depolymerase that directly degrades that K1 capsule. So, a match made in heaven. Now it's time to get to experimental design. So we have a phage that can degrade EPS, but can't make more phage or kill bacteria. So basically, it can only claw its way through the media, it can't replicate. We then have a phage that can't degrade EPS, but it can make more of itself, and it can kill bacteria. And we have a phage that can do both. So additionally, we'll have a phage that can do neither, but I didn't want to waste slide space on that because I think it'll just probably die. Okay. So what do we actually expect to see? The first thing I expect to see, if it can't replicate, but it can sort of use that depolymerase to claw through the matrix, I expect to see it get sort of not super far into the media because it can't get in and then lyse a cell, get pushed out, and sort of work its way down through the biofilm layers. But I expect to see changes in the local matrix architecture near the top. For the phage that can replicate but doesn't have that depolymerase, I expect to see it be able to diffuse through the matrix, but have smaller sort of areas of matrix degradation as it kills cells and leaves sort of these empty holes, but can't directly affect the EPS. And then for the one with both, I expect truly the best of both worlds with far more matrix architecture modifications and these larger holes. So how am I actually going to get this data? So I'll use something called confocal laser scanning microscopy, which basically I can take pictures of it while it's still living. I don't have to kill it and prepare it, so I can get in situ time-lapse photos. And then I'm left with this giant stack of images that are tiny slices of each of the biofilm. I can bring it into the computer and reassemble a 3D biofilm. And what's cool about this is we can pull it into the software Biofilm Q, and we can both build this biofilm and then also do some analysis on the internal structure. So instead of just saying, this one's bigger and this one's smaller, we can get in and see how that internal structure is actually varying. So what do I expect to see? So individual phage traits, I expect to see depolymerase making sort of larger changes in the matrix while replication is still fighting the good fight. We have, then when they're combined, we have a couple options. We have an additive option if we see that it's basically just the effects of both combined, antagonism if they actually do worse together, or synergistic if they're doing better together. And I predict that it's a synergistic. I think that the ability to degrade the EPS will allow the phage to create sort of larger pockets, which then can increase the diffusion of the phage after they replicate, resulting in an overall sort of compounding effect. So aim 2 is I want to track the actual traits that are going on. So to do this, we're going to bring back that beautiful bead method, and we're going to do 24-hour incubation with both the host and the parasite, which should form a biofilm on that bead, and that bead should contain both the bacteria and phage within it. I'm going to collect a little bit of the supernatant, so that outside liquid, just to see what's actually living in that area, but I'll also collect a little piece of the biofilm to freeze. Each day, I will take the bead out and I will wash it off so we just get the really good stuck-on cells and phage, and then put it into new media for 24 hours more of incubation, and then repeat. I'm going to do this for about 30 days, which does require me to come into the lab every day for a month, but that's the price of experimental evolution. And then after that, I will have all of my beautiful little freezer stocks, and then I can start isolating. This is a highly simplified version of what the isolation will actually look like. For the scope of this talk, we're just going to go in general that I will isolate the bacteria from the phage. So once I have all of these time points, I can do something that's called a time shift experiment. So a time shift assay or a time shift experiment is where I take bacteria and phage from different time points and add them together and see what happens. So in this case, if I have my day 0 bacteria, so basically my ancestor bacteria, and I compete it with my phage from a current time point, basically day 0 phage, I can see sort of what my baseline coevolutionary dynamic is for that biofilm degradation. If I then add phage from a future time point, from day 15 or day 30, I can see how that biofilm matrix is changing over time with the phage. So in day 15, same sort of thing, except now we're able to include phage from a past time point, and then day 30, it'll be contemporary, and then 2 past time points. And I plan on doing this not just with day 0, 15, and 30, there'll be far more. And what I'm assuming, what this graph is assuming, is that these are co-evolving, that the bacteria and the phage are just getting better and better and better and better together. So sort of like an arms race dynamic. This might not actually be what's happening, but I'm excited to find out. So what will we learn from this situation? You'll learn if and how much phage are actually modifying the EPS matrix, and we'll learn the evolutionary strategies that occur in co-evolving bacteria and phage populations. I forgot to mention that this will also be in tandem with sequencing, so we'll be able to not just identify who does better and sort of what general strategies are happening, but we can pinpoint exactly what those strategies are. Okay, so why does it matter if the internal structure is changing? Leads me to question 3. So what is the role of niche co-construction in phage-antibiotic synergy? So phage-antibiotic synergy, we'll get there in a second. Just a quick phage therapy reminder. So the idea is that we want to kill bacteria with phage instead of antibiotics. And the reason I have yes and no down here is because we can, but it's not the most effective way to do it. The most effective way to do it is actually to do it with antibiotics too. So that's the idea of phage-antibiotic synergy, is instead of just seeing sort of like, oh, phage does X amount of killing of bacteria and antibiotics does Y, and then together they're X plus Y, we actually see this massive increase when we combine them. The way we combine them matters. There's multiple different sort of headers of phage-antibiotic synergy. Some people apply at the same time, some people do staggered, and that staggered is actually where we see the most dramatic results. So we see that the combination of phage and antibiotics, specifically when you apply phage first, leads you with sort of this more dramatic effect of biofilm clearance. So specifically this paper right here, we see that they found that the structural changes induced by bacteriophage makes this antibiotic more effective. So this basically is sort of what I've been talking about earlier, of does the structure of the biofilm change? And when I saw this paper, I was like, sweet, my thesis has been answered. And then when I actually went inside and read it, there was a lot of gaps that were missing. So while they make a claim that the depolymerase producing phage was more effective on the biofilm, which it was, they don't necessarily look at how the internal structure is changing and separating sort of the role of the antibiotic from the role of the phage. So this is really good evidence for what I'm looking at, and I can't wait to use their methods. So we have evidence that phage change biofilm structure. Why would we keep studying it? Like I said, there's a lot we don't know. So we want to know how internal structure changes over time. Is it predictable? Is increased permeability actually what is causing the antibiotics to be more effective? And how is the internal structure changing over the phage plus antibiotic application time? So the first goal is to just track the changes in biofilm at high synergy time points. So if we have, what do I mean by a high synergy time point? Turns out the actual time when we add the phage is super important. So if we have antibiotics and we add phage and we need to decide when to apply those antibiotics, we can apply them at a variety of time points. A few different studies have done this. These are the most common. People pick 3, 6, 12, or 24. Sometimes they throw 8 in there. And what we see is if we add phage and then we apply the antibiotic and then we have this 24 exposure window, we get some really interesting information. So this graph right here did just that. They're measuring heat flow as a metric of biofilm production, and they did the 3, 6, 12, and 24-hour pretreatments. And then applied antibiotics for 24 hours, took whatever was sort of left in that tube, put it in a new tube, and saw what grew. So what we see is that the 3-hour treatment and the 6-hour treatment, each one of these peaks is basically representing a concentration of antibiotics in that 24-hour antibiotic treatment. And in 3 and 6, we see that it took a concentration of 6 milligrams per liter versus 4. But when we got to 12 hours, we're down to 2. So if we let phage sit on the biofilm for 12 hours before we treat with antibiotics, we can use far less antibiotic concentration to actually get the same result. What's interesting is, so we see the most effective is that 12 hours. But if we jump to the 24 hours, if we let phage sit on for a whole 24 hours before putting on antibiotic, suddenly we've lost all of that progress that we gained. It's suddenly less effective. And what I'm wondering is, what is happening in between here? What evolutionary and structural changes are suddenly occurring between these 2 time points? And is 12 versus 6 the most effective? Can we get higher resolution between these 2 points to figure out what is actually the ideal window? So, aim 1, I'm going to do a similar experiment to what I just showed you, but using sort of a higher resolution and different organisms. I'm going to use E. coli, the same E. coli that I used in the last experiment, just so I can sort of have this consistency. And then I'm also going to add in Streptococcus mutans, which is isolated from sort of a dental disease situation. There's less literature on it, but it's super medically relevant. It's BSL1, and it's different enough from E. coli to provide us sort of this counterpoint interaction. And these are separate tubes. They're not going to be sort of a co-species biofilm. So what I'm going to do is I'm going to grow the biofilms, apply the phage, sort of at this much higher resolution variation. Apply the antibiotics, let it wait, and then do that same sort of tracking of the biofilm, but using that confocal in situ microscopy, and then analyzing with biofilm Q. So after we figure out what the highest best synergy time points are, we then need to figure out, is the antibiotic diffusion actually what's happening? Is the better permeability through the biofilm what's causing this high synergy? So what I'm going to do is I'll take that same sort of experimental design, but instead of applying only an antibiotic, some treatments will have antibiotics, and then I'll have a sort of a paired treatment of a small molecule called fluorescein. This is like an inert, just tracker molecule that I can look at under the scope. And what I want to figure out is, if I think this is happening, where the antibiotics are getting deeper into the biofilm, we should also see that same thing with this sort of inert molecule. But if we see that it's sitting up on the top, that means that something else might be happening, that there's a different sort of evolutionary mechanism that's causing this high synergy, and then we have to go back and take another look. Okay, so what will we learn from this? So we're going to learn what the internal structure of a biofilm looks like at these high synergy time points. We're also going to learn what phage parasitism changes in small molecule diffusion. So it'll be a way for us to sort of build out this idea of if phage are changing the niche and changing it in a way that actually matters for selection. So in summary, we see that phage play a role in modifying the biofilm niche. We see this through the increased production of biofilm. We see this in the change in the amount of eDNA and different like proteins and nutrients that get released when phage are attacking bacteria. We also see that the phage biofilm literature is pretty split. We see a big dichotomy between the evolutionary ecology-focused papers and also the medical literature. We see that the dynamics of bacteria in a biofilm-phage interaction is understudied. A lot of these are eradication motivated, so we don't necessarily know what's happening evolutionarily when a biofilm does or does not become eradicated by phage. And then lastly, we hopefully know that niche co-construction may help explain and optimize phage antibiotic synergy if we actually see this variation in the niche. Okie doke. I want to say my thank yous to my committee, to the Zaman lab as a whole, and a thank you to my family, my friends, to my very first mentor, Josie Griffin, who you can see I only have a photo of her arm here. This was my very first day in a real science lab, and I took this photo to send my parents. A big thank you to my roommates, and then to you, Kai, for both being sort of my partner in crime for these Tuesday seminars, and also for helping me dispose of a chipmunk that I accidentally killed in my garden last year. Questions?
Okay, so you talked a lot about eco-evo stuff. But what about the populations? How are you going to determine the titer or the phage population that you're going to add to the biofilms?
Yes. So I'm going to do a lot of like looking at what other people did and sort of developing backwards from that. I need to figure out first how much of a titer per like the strain that I'm using to like that I'll actually sort of stay in and co-evolved for long enough for 30 days, because the issue of like having your phage go extinct is super common in the sort of like well mixed culture situation. Biofilm, because biofilms sort of act as almost this like slowdown effect, you have weaker selection on the bacteria to become resistant to the phage, so you can end up with sort of these like pockets and reservoirs within the biofilm itself where the phage sort of stay in. So figuring that out is going to be like a whole quest in itself.
I guess great talk. I have more like a comment than like a question. So I was just thinking about when you have phase, you get really cool spatial structures, right? And you're kind of looking at it from looking at the different traits, but it might be interesting to look at this population density, especially because you're going to have like all the time points with the time lapse. So just looking at like lot couple, there are some sort of oscillations and how that might help explain.
Yeah, that would be super cool.
I have like a really specific methods question, but also, like Nikesh said, excellent talk. So for the time shift experiments, which are super cool, just a practical question, like how do you separate the phage and the bacteria so that you can do those shifts? Sorry if I missed that.
No, this is, yes, I have sort of two working ideas right now. So one of them is I could go in, I could do sort of like a standard isolation where I streak out the bacteria, I plate for a phage, and then sort of pick colonies and pick plaques and sort of try to recreate the population, or do like very individualized, like break down individual patterns. Also, I'm trying to figure out how I could sort of maintain that population structure still. So if I think I can filter out the phage from the bacteria. I'm trying to figure out if I can then take the bacteria off like the filter paper and then use that as the starting point for like the bacteria population. So that's something I'm trying to figure out. I was thinking about that yesterday. No.
Okay, so you talked about beavers at the beginning, creating their niche, right?
Yeah.
Beavers are doing a lot of work to create this structure. Are the bacteria spending a lot of energy creating biofilms? Is this a passive structure? Is it an active structure?
Yeah, so it's an active structure. They have to produce those polysaccharides. They have to make those sugars. They have sort of like it's a cost-benefit analysis at the end of the day, where evolutionarily they've sort of like these cellular triggers where it's safer and more cost-effective to spend this time producing a biofilm that you can then sort of hang out in than it is to be a free-floating bacteria where you also then have to try to find nutrients. Biofilms benefit from sometimes you're on a substrate that actually is your nutrient source, or you're in an environment where then the nutrients are sort of diffusing inward through that biofilm at the top through those water channels. So it is expensive, but it also like evolutionarily has become less expensive than just hanging out.
Do you ever find pairs of bacteria and phage that become positive associations? You talked about phage always as negative, something that's either destroying the bacterium or being overcome if the bacterium evolves resistance. I'm wondering, thinking of some niche co-constructions, are there symbioses that ever arise here?
Yeah,
Do you expect to find any in any of the work that you're doing?
Yes, so the parasitism mutualism gradient is very real in this situation. In the specific instances that I sort of pulled out to look at, I'm not sure about if there's going to be sort of an established sort of symbiosis situation. I think that there's a very good chance, because one of the nice things that phage do for the biofilm is they do kill cells, which then pushes nutrients back into the system. So if you are on the brink of populating your biofilm and you have phage come in and then reflush in those nutrients, you can sort of have this situation where you might save your population from starving out. There's a really cool Pseudomonas phage where it's filamentous and they build almost armor around the bacteria. So you see this production of the biofilm with this really incredible lattice structure of these phage that act to actually protect the bacteria. So you get from this almost toxic mutualism to a mutualism. So I'm excited to see where on the gradient this is going to fall.
I can ask a related question. As I was listening to you talk about the experimental design, I was thinking a lot about the question of which phage and does the phage host pair matter and to what extent. So I'm curious, I'm sure you've thought about this because you shared about the 2 different hosts you'll try. Have you thought about different phages on one host? And how does because I was trying to listen, zoomed back, listening from the high vantage the whole talk, and it seems you're aiming to create a framework of understanding how these things are interacting, with the end goal to be can this maybe be applied in pharmaceutical context, PAS thinking? So where in this framework that I think you're developing can you account for changes in phage or changes in host? Does that make sense?
Yeah. Okay, so the first part of the question about which phage and how different phage?
Yeah, just have you considered other phages?
Yes, so this is the one that I found that had the most obvious connection. The K1F to K1 was the most obvious one that was it will directly attack the depolymerase, or it has the depolymerase to directly degrade the polysaccharide. I want to try different phage. We have the whole basal phage collection in the lab, which has a lot of really cool opportunities. I haven't dug into it as much as I want to, but that's one of the things that I want to look into on, even if it doesn't have this depolymerase, what kind of phage strategies are out there that still could predate these or parasitize these E. coli. Second, inspired by Megan, their lab manager in our lab, the phage cocktail sort of work of how you can sort of do phage cocktails and do phage training in a well-mixed culture. What kind of if you suddenly, instead of just one parasite strategy, you have 20, what happens? How does this change sort of the internal structure? It can sort of build very quickly because you can have multiple strategies within a biofilm because they're so spatially structured and heterogeneous that you might have a bacteria over here that's really good at defending itself from one phage and not one on the other side there. But if they just never happen to interact, suddenly you don't see that relationship come up. So I do want to sort of build up these multi-phage experiments eventually, yeah.
Yeah, that's kind of what I was thinking with the framework question, because it can obviously exponentially increase in complexity. And so maybe there's a way to tackle it from a logical perspective of when does it matter how robust this is to different numbers of phages or different numbers of hosts, and then kind of designing the theory or framework around that. When does it matter?
Yeah, the phage antibiotic synergy part, I'm gonna flip back to this graph right here, this switch. This is what I think I'm most interested in the context of when does it flip, when does it matter, when do hosts and parasites change, is I presume basically something happened evolutionarily between 12 and 24 hours where the bacteria, maybe that's when they started producing way more biofilm, the phage can't handle it, they basically become ineffective and or no longer, that area is no longer as permeable as it once was. So when you apply antibiotics, you see all of these heat spikes again because you see no matter how much antibiotic you apply, you end up with biofilm growth anyway. So I think keeping track of that and doing a lot of evolve and resequence work is going to be important to make sure that I'm not missing that gap.
First of all, that was a really great talk. I'm curious about more about the structure of the biofilm and specifically about the extracellular DNA. And I'm wondering where it comes from and if we know if it does anything else besides provide structure.
Yeah, so comes from mostly the dead cells in the biofilm. So like when a bacteria dies, they don't like shove it out. They sort of just recannibalize it, and that DNA ends up getting trapped in the sugars. There's some interesting, I think this is more bacterial, just like debris in general, where you can see phage that sort of get trapped because they find the little like protein that they normally grab onto, but it's just like a little chunk of bacteria corpse, so they don't actually do anything. So like that increasing cell death rapidly also leads to like maybe a less effective phage population, because now you're more likely to grab onto a receptor that doesn't actually lead you to an infection cycle. So I think sometimes they can attach to EED or to DNA. I have a vague memory of a paper that said that, but I'd have to go check.
That was an amazing talk. I love all the figures. They were lovely. I was thinking, like, do you expect to see that, like, viral debris, whether that be the DNA or proteins, contribute to the biofilm structure?
Ooh.
Like the capsid proteins or the tiny chunks of DNA?
I don't know, but that's a good question. Yeah, I think a lot of the next sort of thing I want to develop is figuring out what I'm actually going to do, like staining and tagging for the confocal. So I think trying to figure out a way to do live dead staining on bacteria, but also if there's a way to do live dead staining on viral particles. Yes, I'll get back to you on that. Because viruses are alive. Hot take.
Otherwise,
Megan has a question.
Megan.
Megan?
It's the same question.
Ooh.
Okay, Lisa was asking that in the studies that were found 12 hours were the sweet spots, did they use the same bacteria you plan to use in your experiments?
Hi, Auntie Lisa. No, it's not. They didn't use the same. They used much scarier bacteria. So they used Pseudomonas aeruginosa and MRSA, which, Luis, do you want to bring some MRSA into the lab? I'm getting a no. No, they use different bacteria because this is a hardcore medical eradication study. But that is like a good question of how similar do these bacteria act? How similar do their phages act? And that's something that I think is really interesting, jumping back to that split in the literature, taking an eradication approach with a non-sort of escape pathogen. Do we see the same thing, or should we really be tailoring our approaches to pathogen specific?
Great job.